The pathology of sarcoidosis centers on a control mechanism that has failed. It is an immune response where the structural off switch has physically jammed in the on position. In a healthy response, a macrophage engulfs an environmental antigen and presents it to a T cell to neutralize the threat. Once the threat is gone, the system achieves immune tolerance. The cells detach, the inflammation resolves, and the system powers down. In sarcoidosis, the immune system fails to execute this shutdown. We can map this failure starting with an environmental trigger, like a bioaerosol. While usually harmless, in a host with specific HLA class II alleles, the immune system mounts an exaggerated adaptive response. Instead of standard clearance, the macrophages trigger a massive Th1 response. This activation releases the cytokines IFN-gamma and IL-2. These act as igniters, commanding the T cells to multiply through rapid oligoclonal expansion. This explosive growth marks the point where the response deviates from normal clearance into an exaggerated antigen-driven state. Those igniters then transform the macrophages into epithelioid histiocytes. These cells begin releasing builder cytokines, primarily TNF. TNF recruits more cells to clump tightly together, walling off the perceived threat inside a non-necrotizing granuloma. Building this granuloma is a standard containment strategy used by the body to wall off threats. The clinical disease begins when this wall fails to dissolve. Defective regulatory T cells and excess TNF allow the macrophages and T cells to continuously activate each other. They form a self-perpetuating inflammatory circuit. Look at this specific circuit. If you are a rheumatologist looking to break this feedback loop, which cytokine node would you target? There is a short timer on screen if you want to pause and think. Targeted anti-TNF therapies work by striking this node. By severing this feedback loop, the medication allows the persistent granulomas to finally disperse. This infinite inflammatory loop defines the disease state. While granulomas appear in many conditions, the sarcoidosis pathology is driven by this specific failure of the feedback circuit. If the inflammatory loop runs for years, it awakens fibroblasts via the cytokine TGF-beta. This process turns the cellular wall into rigid, permanent scar tissue. This fibrosis leads to architectural distortion and irreversible organ damage. This immune mechanism remains the same regardless of location. The process is identical whether it occurs in the lungs, the heart, the eyes, or the nervous system. This is the complete flowchart of the disease process. You can see how the initial trigger leads through the loop and into final fibrosis. This is a good moment to take a screenshot for your notes. Every clinical stage stems from this single architectural flaw. Sarcoidosis is the chronic failure of immune resolution in a genetically susceptible host.